So one of these then important functions of DNA obviously is to code for protein synthesis. And we used to have this concept of DNA as or of genes as a part of DNA that codes for a particular protein. And so there would be a gene for each of the proteins that's critical in your body for your body to be able to function. And this has kind of been thrown by the wayside as we've done more and more research into the functions of certain genes, because there are some proteins that require the action of 4 or 5 different genes, some that require the action of many more than that. So we had to redefine genes as perhaps only coding for a portion of a protein instead of the entirety of a protein. We also have genes that code for RNA. I mean, that's how we get the RNA that's present in the nucleus for DNA transcription. We have genes that code for regulation, basically. We've got genes that basically determine the code that embryonic development is going to follow. There's no end point or end product like a particular protein, but rather that gene may say, if expressed in the 8th week of development or the 10th week of development, then code for limb buds. If expressed during the 20th week of development, do this different function. So we've redefined genes as a sequence of DNA that specifies the sequence of amino acids in a particular protein, that specifies the sequence of amino acids in a portion of a protein, or that is responsible for any functional product, including RNA. And so a particular gene may be made up of hundreds or even thousands of DNA molecules, of nucleotides, organized into both coding and non-coding segments. And so as transcription happens, both coding and non-coding segments are copied. As translation happens, and this sequence of RNA that comes out of copying the DNA is then processed and formed into proteins, there may be parts of the copied DNA that are cut out or excised. And so we distinguish between the exons, the non-coding regions, and introns. Our genome is defined as the entire genetic makeup of an individual or species. This includes our 25,000 functional protein genes, as well as any spacer DNA, any non-coding DNA. All of it is our genome. The exons are the segments of genes that are transcribed and then involved in protein synthesis. Non-coding DNA does not direct the production of proteins, but may produce other important molecules. There should be a Y on may. I'm sorry for that misspelling. Introns used to be considered synonymous with non-coding DNA, but introns are segments of genes that are transcribed and then deleted. They can vary from protein to protein. So if the same gene is involved in the production of multiple proteins, sometimes that particular segment that's copied may be an intron. That is, it's deleted out in the process of translation. But for other proteins, that segment of DNA may be kept as an exon. So as we understand more and more about the structure of DNA and the structure of genes and how these genes are then transcribed and kind of processed or translated, we can see that the same basic sequence of genetic code can serve multiple functions depending on the regulator that is kind of controlling the expression of that particular gene. And so here this diagram demonstrates translation. We have DNA with its segments of both exons and introns during transcription. Those introns might be cut out, where primary messenger RNA then codes only the exon sections, and then during the mature RNA travels to the ribosome and is then translated into the end product or the folded protein. So Some genes don't produce anything. Some genes don't have a functional product that is produced, but are very incredibly important genes nonetheless. We call these genes regulatory genes. Regulatory genes are genes that control the expression of other genes rather than producing some end functional product. The products of regulatory genes are maybe enzymes or other kinds of hormones that may mediate the expression of other segments of DNA. And so there's one particular kind of regulatory gene called homeobox genes that are critical. Our Hox genes, for example, direct the early segmentation of embryonic tissue. They also interact with other genes to determine the characteristics of the developing body segments and structures. So Hox genes determine where limb buds will appear. If they're messed up, you get a limb growing out of your back or growing out of your face. Also, things like how many and the pattern of the vertebra that develop. We've got thoracic, lumbar, and the I'm spacing on the term for it. The thoracic are your neck vertebra, the lumbar are your back vertebra, and then we've got those that are part of the coccyx or the tailbone. That's what I can't remember the term for. But anyway, these three kinds of vertebrae have very different shapes and go together in a certain sequence to form the total functional spine of an organism. So these Hox genes that regulate this sequence of development. The lumbar or the lower back. It's thoracic, which are the ones in the upper back. These Hox genes that regulate this kind of pattern of cell division and cell differentiation are critical. If we didn't have reliably copied Hox DNA, if this wasn't something that was preserved or conserved through evolutionary history, we would have incredible problems that develop in the developing embryo. So these Hox genes and other homeobox genes are highly conserved. We don't see a high mutation rate of regulatory genes, particularly this kind of regulatory gene, because when we do have mutation in these genes, we end up with very far-reaching developmental abnormalities. We end up with birth defects that may be incompatible with life, like anencephaly or the lack of a brain, or these neural tube defects where the spinal column or even part of the brain form outside of bone, etc. Changes in the behavior of homeobox genes are responsible for a lot of the physical differences that we see between closely related species. So if you recall, during the introductory lecture, I talked about how Sulawesi was the hotbed of macaque evolution. Well, one of the things that varies between these very closely related species of macaques are the patterns of their sexual swellings. It's regulatory genes that lead to these different swelling patterns, not genes that are coding for a final product that's a functional protein. Or even between breeds of domesticated species. So the reason that Great Danes look functionally different from Chihuahuas, or that pugs have this messed up face, these are all due to homeobox genes that differentiate this pattern of early development between these breeds. And so we've done similar sorts of artificial selection in our cereal grains as well. It's homeobox genes that are implicated in turning corn, turning teosinte, or the grass that corn came from, into full-fledged modern corn. So these regulatory genes are critical. They are things that we can manipulate for specific desired outcomes in this process of artificial selection, but thankfully in the process of natural selection, they're pretty highly conserved, so that most of the embryos that we form through sexual reproduction are going to develop properly. They're going to develop along the right kind of sequence of events. Genes are organized into chromosomes. Chromosomes are discrete structures composed of DNA and proteins that are found only in the nucleus of the cell. They form when DNA condenses and then becomes tightly coiled. This is in that very first kind of component or stage of cell division. During cell division, chromosomes replicate into two strands that are joined at the centromere. So what happens during DNA transcription during the process of cell division is that DNA is copied, that strand is copied in its entirety, and this results into homologous chromosomes joined at the centromere. Our autosomal chromosomes carry genetic information that governs all physical characteristics except for our primary sex determination. Every cell in our body has a full complement of autosomal chromosomes, even our gametes. Every cell in our body also has a copy of our sex chromosomes, but based on where those cells occur, different genes on different chromosomes are going to be activated in this process of cell and tissue differentiation. So you can, for example, take a skin cell, an epithelial cell, and sequence an entire genome. It is entirely possible. But if you are determining sequence through the expression of the genes in a particular cell, you're not going to get a full picture of what the entire completed organism is able to do, because in epithelial cells only some genes on some chromosomes are expressed. In neural cells, some genes on some chromosomes are expressed, so on and so forth. Sex chromosomes carry genetic information that determines maleness or femaleness. In mammals, females have two X chromosomes, males have an X and a Y. In birds, we've got Z and W chromosomes, and females are ZW, while males, I believe, are ZZ. So we don't see across taxa that females are always homozygous and males are heterozygous. However, we're interested in primate and then human evolution, and so because we are mammals, we are going to talk about X and Y as specific sex chromosomes. The X doesn't really do anything, to be honest. I mean, it does a whole lot. But because X's are present in both males and females, it does a whole lot for both sexes. It is the presence or absence of the Y chromosome that determines primary differences in sexual anatomy. And in particular, there is a gene on the Y chromosome called the SRY gene, or the sex-determining region Y gene, that starts that process of sexual differentiation in the developing embryo. And so at about the 5th or 6th week of gestation, if there's a Y chromosome with a properly functioning SRY gene, it will start a cascade. It'll release a surge of testosterone that starts a cascade by which the Mullerian ducts of the totipotent fetal gonad are disintegrated. The Wolffian ducts that become the testes and such proliferate. We get the testes, the vas deferens, the seminal vesicles, etc. We then get also the development of this primordial fetal phallus into specifically a penis. If there is no sex-determining Y gene, meaning that the Y chromosome is not fully functional, may have a mutation that eliminated the sex-determining region Y gene, or there is no Y chromosome, that is, there are two X's present, then the normal pattern of development, or kind of the default pattern of development, is femaleness, whereby the Mullerian duct system differentiates into the ovaries, the fallopian tubes, the uterus, the Wolffian ducts deteriorate, and the primordial fetal phallus becomes a clitoris. So the sex chromosomes, it's that Y chromosome that has a critical role in altering what we would call the default kind of expression or default pathway of development in embryos as they develop into fetuses. We then have processes. I mean, one of the key critical reasons for DNA replication, of course, is for cell division. This is a critical process of both growth and development while we're laying down fully functioning organ systems. While those organ systems are getting larger, our bones are enlarging, our muscles are enlarging, our skin has to enlarge to cover the increased surface area of bones and muscle. It also happens just during what we would consider maintenance. And so we constantly have cells that are dying. And our health and our vigor, our low rate of senescence at younger ages, is due to DNA repair and cellular regeneration. So as old cells die or are injured, new younger cells take their place, and each cell has really a kind of predestined period of life. It's just that when we're younger, we generate many more new cells to replace those that die off. Part of our senescence later in life is because we don't replace cells that are dying off. The process of mitosis is cell division in somatic cells. The DNA replicates. You get these two homologous chromosomes joined at the centromere. The cell pinches in and cleaves between those two homologous chromosomes, and you end up with two genetically identical daughter cells. Meiosis is slightly different. Meiosis is cell division in specialized cells, critically the ovaries and the testes. These are the cells that produce gametes. And meiosis involves two phases of division that result ultimately in four daughter cells, each that will contain only half of the original number of chromosomes. And so in meiosis, you get the DNA replication. You have your homologous chromosomes lining up on either side of the centromere. You first then cleave into two cells that contain one set of homologous chromosomes. Then you cleave into four cells that each contain only one chromosome. And so sex cells ultimately have half the number of chromosomes that our autosomal cells have. So since we have 46 chromosomes in 23 pair, each gamete is only going to contain 23 chromosomes. So one other thing to just keep in the back of your mind about gametogenesis, and it's not something you're going to be tested on from this week's module. It just carries over into our future discussion of how environment interacts with our genome to produce a lot of the measurable variation that we see today. Your mother, the process where her ovaries developed the gametes that gave rise to you, happened while your grandmother was pregnant with your mother. So female gametogenesis in the ovaries happens in utero, meaning that while we are developing in our mother's uterus, that's when our ovaries are producing all of the gametes that we will ever possess. So it adds an additional level of complexity to the way that environmental and genetic factors kind of interact. Males undergo gametogenesis in their testes their entire adult life. It starts usually between the ages of about 8 and 10. They start producing sperm well before they ever start this process of growth and the development of secondary sex characteristics like facial hair and upper body strength. So there can be a different kind of more immediate or proximal role, perhaps, that the environment plays on gametogenesis. You get a blast of radiation, right? As a set of gametes are being formed, they're probably going to have a lot of mutations. Hopefully, the cells that generate those gametes didn't, and it's just like a batch or two of sperm that you have to pitch. I mean, that's one of the roles that, or one of the effects of chemotherapy, is that chemotherapies cause mutations in cells. And so if you're not storing let's say you have to undergo treatment for cancer, and you're not banking up sperm before the chemotherapy is ever done, you're not going to save any of the sperm that you would ejaculate during this process of your chemotherapy treatments, because those cells are going to be more likely to harbor mutations. Meiosis is pretty critical in the development of higher taxa, because it is through meiosis that we allow for sexual reproduction. I mean, if we're just talking about mitosis, we get identical daughter cells from cell division, and this is essentially what happens in asexual reproducers, that they develop eggs that have the full complement of all of their chromosomes, and then those eggs develop, hatch, whatever, without any influx of new DNA. So those resultant daughters are genetic copies of their mothers. I mean, this is how all the sisters in a hive, a beehive, or an ant colony are going to be genetic copies of one another because of this role of mitosis in asexual reproduction. Well, birds and mammals can't do it. Right? Birds and mammals cannot reproduce asexually. So, somewhere along our evolutionary history, the benefit of sexual recombination outweighed kind of the cost of sexual recombination. I mean, when we have sexual reproduction, your offspring are only half related to you, right? 50% related to you, because half of their genes come from their fathers. And so this is one of the things that's a component of what we call the twofold cost of sex, that we basically have to produce twice as many offspring that are going to survive and reproduce to have as many copies of our own genes out there. So why? Why are we willing to bear that twofold cost of reproduction in order to have sexual recombination? Well, sexual recombination in higher taxa produces really important variation on which natural selection can act. This is of critical importance when environments are unpredictable, particularly when you have no idea what the future is going to be like. And so one of the areas of our evolutionary history where this unpredictability is forefront or is of key importance is the evolutionary arms race that we have with pathogens. So what does this mean? Well, there's that scene from Alice in Wonderland, and then Richard Dawkins borrowed it for his book, The Selfish Gene, where he talks about the chapter, he wrote a book called The Red Queen as well. And that scene where Alice is racing the Red Queen, she's running as fast as she can, not to get ahead of the Red Queen, but just to stay even with the Red Queen. And that's kind of what we are doing with pathogens. We are evolving our immune systems absolutely as fast as we possibly can, only to keep up with pathogen evolution. I mean, microbes, bacteria, fungi, viruses, they can evolve and mutate at rates that are so much more dramatic than ours because of the fact that their intergenerational span or time is so much less. And so they can go through thousands to hundreds of thousands of generations in the time that we only produce one offspring. So in that respect, we can't predict what our offspring's pathogen load will be. We can't predict what kinds of immunocompetence our offspring are going to need, and therefore getting a lot of variety through sexual reproduction is really, really important. And we actually find in habitats that have a higher endemic pathogen load, so tropical environments, there's a lot more of what we would consider infidelity. That is, you don't see pair bonding and monogamous mating. You see mating with multiple individuals to up the variation in your offspring. In environments that are colder, that are higher latitude, because the pathogen load is a lot lower, we are more likely to see monogamy. So sexual recombination is critical. And this is one of the roles that hormones play, right? We talked about the role of pheromones and attractiveness. This is why we smell people differently. It's because we are looking for that immune system that is similar enough to ours, but different enough from ours, that we are going to generate offspring who have a new novel immune system that is really, really well suited, really, really functioning, high functioning, really, really healthy.